Hi, my name is Sarah Saldana, and today's date is February 4th, 2018. And in this video, I'm going to be going over the process of the biochemical testing experiment that we were supposed to do this week, and some of the results that should have happened. So in this experiment, we went over information pertaining to the methyl red test, which is normally done hand in hand with the Voges Proskauer test, as well as Catalase testing. So these three tests mentioned are used to identify different microorganisms. The methyl red test is used to identify those that are involved with mixed acid fermentation. It's a process that requires no oxygen but produces lactic acid, acetic acid, ethanol, carbon dioxide, and hydrogen gas. Cultures that take part in mixed acid fermentation are mixed with methyl red, and those that are positive turn red, those that are negative don't. So they turn more of like a yellow color. That second test, that Voges Proskauer test, it's similar in that the substances are added to cultures that have been incubated in the MRVP broth, but instead it differentiates or it identifies microbes that ferment glucose by means of the butanediol pathway. So another difference is that this test does not use methyl red, but instead it uses chemicals known as Barrett's reagents. And then for that third test, the catalase testing, it's used to observe the breakdown of hydrogen peroxide into oxygen and water in catalase positive microbes. So this test is also in addition for aerobic microbes, so microorganisms that use oxygen, and hydrogen peroxide results in bubbles that form rapidly, so that is a positive result, and then those that lack bubbles when added with hydrogen peroxide yield a negative result. So the reason I'm explaining this instead of showing it is because unfortunately this week I mixed up materials from this lab as well as another lab assigned this week, so I was not able to perform the experiment, but I did want to go over some of the results that I would have gotten had I been able to complete it. So to go back to that methyl red test, using methyl red dye, E. coli, and S. epidermis, for the E. coli sample, I would have received a positive result, so that culture in the MRVP broth would have stained red when added with the methyl red, because it's positive for acidic products after the solutions combined. As for the second test, the Voges Proskauer test, the E. coli would have turned copper color, providing a negative result. However, for S. epidermis, in the methyl red test, that actually would have proved to be negative, so it should have turned more of a yellow color than red, or instead of red. And for the Voges Proskauer, it would have been positive because it does ferment glucose. And lastly, for that catalase testing, both E. coli and S. epidermis, when added with hydrogen peroxide, should have produced bubbles, so it would have turned into water and oxygen, proving a positive result for the catalase testing. So again, I was not able to complete the experiments this week, but those should have been the results that I would have gotten had I been able to.